Okay, I think it's now recording. Okay, so hi everyone. I'm a postdoc in Luke's lab, and then I will present some of the basic stuff about the spike sorting, and then later the Ellie will show you some demo about how you do spike sorting using the cluster kilosort. So I need to share some my share the screen. Okay, whiteboard. What? No, yeah, this one. Okay, share. So everyone can see my slide. Okay. So I will just touch basic stuff about the spike sorting. Spike sorting is just the grouping the spikes into the cluster based on the similarity of the spike, spike of their shapes or features, and then the goal is to determine which spikes corresponding to which of the neurons, for example. So if you record large number of neurons simultaneously, you wanted to sort which spike is corresponding to which type of neurons. Then why we need the spike sorting? I think it's basically the same question as why we want to have a large number of neurons recording at the same time. I think that it gives a lot of benefit. One main purpose of having large number of neurons recorded at the simultaneously is that brain processes are reflected by the activity of large neuronal populations, and the study of single cells in isolation gives only a very limited view of the whole pictures. And not only that, I think if you record large number of neurons simultaneously, you can address different questions than the single cell recording. For example, you can looking at the connectivity between different neurons, and also you can record several different brain areas simultaneously. And also for single cell recording, it's biased toward to the high firing rate cells, but in large number of cells simultaneously recorded, we can see like sparse spiking cells, and then see what this neuron are doing or encoding, basically. So this is a picture, just an example picture of the neuropixel. I think Luke mentioned about these neuropixels. It's basically the one single probe that has like thousands of sites of recording, and then it expand it to the 1 millimeter. And you can record 300, for example, 80 sites simultaneously. So you can see that this is basically the low data of this neuropixel, and then you can see that there are some cells we're detecting here, and then it's just x-axis is the depth of the probe, and then y-axis is the time. So basically you can record many neurons simultaneously. Before going into the spike sorting algorithm, or like how it works, I wanted to show some data that I took. This is just the silicon probe. So we call this silicon probe, in the silicon probe, we call one probe, one like this mark, one, how can I say, electrode is a shank. So this probe has a 4 shank, and each shank has, for example, 8 different sites of the recording, and we call it the channel. So it has 8 channels in each shank. So it means that it has 32 channels. And I inserted into the mouse hippocampus. And this silicon probe is good in a way that you can record many neurons in one particular region. And then this is basically the bottom figure is basically the raw data that I took from the hippocampus. So you can see that there are slow theta oscillation about 8 Hertz. So you can see that there's a wave. And then on top of that slow wave, you can see that there are some spike downward. That's the spikes. And then the goal is like how you're gonna isolate how many neurons are here, and then whether you can isolate the spikes into different cluster into different neurons. So before going into the silicon probe, so let's go over some basic spike sorting method. One just simple method is if you look at the low data, and then you just do some kind of amplitude threshold. For example, you just cut like high amplitude one neuron and the low amplitude the other neuron. That's very simple way, but you can see that this is not a right way, and then also it's bad, poor quality of the clustering. And one like method that people are using and they commercially available, and also like a lot of people still are using this method, is a window discriminator. I used this method before because I use like 3 to 5 electrode, tungsten electrode, and then put insert into the brain, and then do online sorting. Online sorting meaning is that you sorted the cells while you are recording. While animal is behaving or while you are recording, you can sort the neuron. Basically, you put some kind of window to discriminate the different cells. You can see that there's a blue window to capture the blue, the spike that colored blue, and then the red window is capture the cells that has a red labeled cells. It's convenient and also it's easy and intuitive, but there is some drawbacks. For first, I mean, you have to manually set this window by user, and then you can do only like few things at the same time. And the other drawback is that some cells having very similar feature, so that it's hard to distinguish between different cells. So, and then also if you have a silicon probe, I think it's not easy to do it because it takes a lot of time, and then you do have to manual setting of the window by yourself. Okay, so then what is the basic step?
Can I ask a question?
Okay.
About the previous slide? Mm-hmm. When you set the window, is it literally you just like draw that window, and then if it falls, if the signal falls in that window, then you call it?
And then, that window, whatever that, for example, blue, whenever it has these two blue window, that considered as a one cell, blue cells.
Okay, so it has to pass both windows, and then you call it. Okay, got it. Thank you.
Okay. And then this is the basic step for, I mean, first so let's just simplify, not using the silicon probe, but using just tungsten wire. And then this is the shape of the brain, I mean, the region of the brain. And then you put the electrode, and then in the figure, you can see that zone 1 and zone 2 and zone 3. Zone 1 is basically near the electrode, so that you can isolate the cells because it's close, so that you can see like very clear shapes and amplitude is big. Zone 2 is basically, it's close, it's not enough close so that you can use, you cannot isolate it, you can have like multi-unit activity. And zone 3 is basically, it's background noise. It's too far so that it's detected as a background noise. So it's a middle figure is the low data. And then when you do low pass, when you apply the low pass filter, then you can get local filter potential. And if you use high pass filter or band pass filter around like 300 Hertz to the 4,000 Hertz, then you can have bottom figure that has flattened, but having some kind of the peak of the spikes. And what you need to do is you have to detect the spikes and then sorting according to the shape or the feature of the this spike, the spikes. So I will go more detail in the next slide. So this is basically basic step for the spike sorting. Low data, and then next you filtered, high pass filtered, so flatten so that you don't have any kind of slow oscillations. And then you do spike detections. It's spike detections, and then go to the feature extraction. It's basically dimension reduction, so that I will explain more detail in the later, and then do clustering, and then you cluster two different cell type. So as I said, you first filter the data. After you filter the data, we have to detect the spike. So the basic, like common method to detect a spike is the amplitude threshold. So that you just put the threshold, so for example, red or green in the channel 1, then you can just threshold, and then whatever the past the threshold you consider as spikes. So, but you wanted to get, you wanted to have amplitude threshold so that you don't want to missing the spike. If the threshold is too high, then you're gonna miss a lot of spikes. If you put too low a threshold, then you're gonna getting the false positive, so that you can get a lot of noise. So you wanted to have a proper threshold to detect the spikes. So commonly used method is using some kind of standard deviations. By using standard deviation gives some kind of, how can I say, I'm sorry, some kind of different threshold for different channels. For example, if you look at the channel 4, that this is more, how can I say, the activity, there are not much activity, so the standard deviation is low, so that you have low threshold. But in channel 1, you have a lot of spikes. Because of these spikes, you have higher standard deviation of the activity, so that your threshold is high, like green, so that you miss a lot of spikes. So one way to avoid this is to use some kind of median method. So using some kind of activity, median activity, and then use the threshold, that's the red, so that you can have low enough to detect all the spikes and high enough to get rid of noises. And then, so this red is using the median method, and then the green is basically using some kind of standard deviation method. Is that clear, everyone? Okay. Okay, then after...
Can you make your slide a full screen or no?
Oh, sure, sure.
Thank you.
Sorry, the reason that I didn't do slideshow is because, I'm sorry, because for some reason my computer like crashed when I do the slideshow without putting the output. I don't know, I have to fix it, so that's why. Is it good enough?
Okay, it's fine, otherwise it's good enough, yeah.
It's good enough, okay. Sorry about
that. Can I ask a question related to your previous slides?
Okay.
No, the previous one. Yes, actually, when we need to low pass filter and when we need to high pass filter, I didn't get it.
Oh, I see. So basically, this one is that. So this is I don't know whether you cannot see my point, right? Pointer. So you have a low data, and to get the low oscillation, you need to do low pass filter. So only low frequency is passed, so that you can have, like, for example, theta or gamma oscillation is detected if you low pass filter, and high pass filter when you wanted to detect the spike. Okay.
Yes, thank you.
Okay, and the next step is after you extract the spike time, the next step is feature extraction. This is, I think, most challenging, one of the most challenging part. Basically, after you, so this is the previous figure showed that you high pass filter and then you have a spike and then you wanted to dissociate, I mean, cluster the cells, cluster cells depending on like the shape of the spike shape. And it's basically, for example, you have a sampling rate of, so you're taking a data with a sampling rate of 30 kilohertz. And then you want to have, once you have a spike time, you want it to have a time window that capture the spike waveform, for example. And then spike waveform, for example, lasts for a reasonable amount of time for 2 milliseconds. So 3 kilohertz sampling rate, and then you want to have a 2 milliseconds for each spikes, then you have 60 data point, right? So you have a 60 data point, and then you wanted to reduce it to like, so meaning that you have a 60 dimension for each spikes, and then you wanted to reduce that to like 2 dimension or 3 dimension based to extract the feature and then separate the spikes. So the basic, the one simple way, I just gonna show you. One simple way is basically just using the amplitude. For example, in the high amplitude one considered as one spike, and then low amplitude one was considered as one spike. But you can see that the green cells and red cells has a very similar amplitude, so that it's not easy to separate those two cells. So you need other features to extract these two neurons. And also amplitude, you can see that in the bottom figure, this x-axis amplitude of each neurons. And then you can see that there's a background amplitude, and then neuron 2 amplitude and neuron 1 amplitude. If you set the background, the threshold of having neuron 1 into B threshold, and you're gonna miss a lot of spikes in neuron 1. And then if you threshold set to A, then you're gonna contain a lot of spikes from neuron 2 into neuron 1. So there are different feature, different things that happening in this feature extraction, spike sorting. So let's look at some example. So for example, this is basically the top left one is basically the one that isolates well cells. So basically you threshold the amplitude, the peak amplitude, and then this is isolated well. But the other example is the right next to the left figure top. You can see that it's thresholding the top, but you can see that, clearly see that you have two different cells. So you can see that this cell, the second cell has some kind of width is larger, and it's slowly decaying cells. So in the left bottom cells, you just plot these two cells spike into the spike minimum versus spike maximum amplitude. And you can see that these two cells are not that different, right? Because if you look at the spike amplitude, peak amplitude, and spike minimum amplitude, this is not that different. But if you plot it between the spike width versus spike height, then you can see that it's kind of well isolated, so that you can see the rectangular shape is like spike 1, so that it's spike width is smaller, and then the other one is spike width is higher, and then a bit amplitude is a bit lower, for example. So this is basically how you extract a feature and then cluster to the different cells. But one drawback, this one, is that you have to look at individual spikes or shape of the spikes, and then see which feature you wanted to use. And then it's very time consuming, and then also it's not well, it's not it's not an optimal method to use this. So what people are using is basically features that the feature extraction using the PCA analysis. So it's basically choosing the feature based on the principal component of the spike. And okay, so it's basically this. The idea behind principal component analysis reordering the axis, so that find find the ordered set of orthogonal axes that capture the direction of the axis that has largest variations, for example. So this this figure shows some example of the two different cells centered on the peak. And you can do some PCA analysis and find it's basically the just transformations, linear transformations, and then you find the axis of the most the variation is highest. To find the axis that has the highest variations. And if you do the PCA analysis, you have different the pieces. You get the different pieces, and then the first pieces, the first component, principal component, is the axis that has the largest variations, and this is first principal component, and second principal component, and third principal component. And then if you product this first principal component into the each spikes, then you can get this this dot for each spikes, and then you can have a first component score, and then you can plot the first component score versus the second component score. And you can see that the, I don't know how to say, but you can see that in the last right figure, you can see the three cluster. And then right top figure is basically, right top figure is basically have a positive first component, and then positive second component is basically the one that has, it's hard to say, but has the highest peak and then the lowest peak. That's the cells that captures. And then the other cluster is the second cells that has the slowly recovering cells that's captured in the bottom. And then the leftmost cluster is basically the noise. So you can separate these different spikes into the different cluster. And
sorry,
then,
I have a question.
Okay.
Sorry, again, it's related to previous slide, that how we can actually determine the threshold. It's just by trial and error, or there is, I mean, a principle to find this threshold.
What do you mean? What was the question?
I mean that, for example, if we want to have this kind of, I mean, spike, we need to have, we need to put the threshold in a, I mean, below, or I mean in upper places. How we can find this is, for example, the threshold should be in this place.
I see. In this figure, you mean? So
yes.
This one is basically the you just set by looking at it, and you just have to set it manually. But for the PCA, so that's why it's not an easy way. I mean, it's not a efficient way to do it. By looking at it, and then you just set the threshold. That's it. But if you use the PCA analysis, they will automatically find the axis that gives you your highest.
So that is the main things that PCA is doing for us. Yes.
Yes, I mean, it's finding finding the axis that gives the largest variation, so that you can separate the cells more better.
Thank you. Thank you.
And then you can use, I mean, it's basically this feature extraction is basically some kind of dimension reduction, process. So you can use different algorithm using like wavelet transformation and other things, but I will not go over to detail about this. But anyway, basically you wanted to reduce the dimension and then find the feature that separate the cells best. Okay, so once you find that feature, you want it to cluster. Like clustering meaning that you want it to separate the cells into large number of spikes into different cells. The one simple way is k-mean clustering. So clustering means that group spikes with a similar feature into the cluster and then corresponding to the different neurons. And for example, if you plot principal component first principal component and second principal component in the previous figure, and then you calculate whether these clusters are close together, and then calculate it together, and then just divide this into like 3 different cluster. It's basically k-mean clustering. This algorithm, it's Euclidean distance measure, and then just calculate the closest, calculate the cluster, the spikes that has a closest mean, for example. And then you have other method of, it's basically the same. You want it to have some kind of distribution that has a multimodal, so that you can separate the cells into the population, the different population into the different spikes, basically. And then you have, for example, Bayesian clustering and k-mean clustering. It's all the same, that you want to have a multimodal distribution in the spike, and then you want it to separate those cells. And then some of the clustering algorithm has a template matching. Is basically once you cluster using, for example, k-mean, you have a mean spike shape, and then you go back to the second step, for example, the filter steps, and then just go through it to see whether you can find the similar pattern with this template, the template that they have, and then capture the missing spikes that match this template, so that it's just iterative algorithm, so that it just go over and over to see whether you can capture more spikes and then cluster together. That's the main idea. And do you have any questions about clustering and feature extractions? Okay, so these are basically how you do the clustering. I will show you some example that I took from the hippocampus. And then this is the hippocampus neural data. And then you can see that this is basically the one shank that has an A channel. And then this is the low data. And then this purple and orange and yellow, the green trace is basically the cell that are clustered into the different neurons. For example, so it's taken from the one shank, and then each line is from the data from one channel, one site. So we have 8 different sites in one shank. And then the first cells has the spikes into the channel number 5, 6, and 7. And then second cell has the highest peak in the fourth channel. And the third one has the highest peak on the fourth channel too. And then if you plot this feature into, for example, channel 4 amplitude and channel 6 amplitude, you can identify the orange cells and green cells and purple cells by looking at it. So comparing to the previous slide, this one, like the silicon probe, has a benefit that you have the dimension that has a spatial dimension, for example, so that each cell can be captured by different sites because it's close enough, so that it has a spatial component so that it's detect cluster cells better, okay? Okay, and then the final step is the manual clustering. I think it's basically you have to go over the automatic cluster and then see whether it's a reasonable cell. I think the Ellie is gonna go over it. And then Ellie, are you gonna go over this problem of spike sorting?
Yes, but I mean, you can show now.
Okay, I mean, if you're gonna show it, then I will not.
Okay.
Okay. Okay, that's it. So I just wanted to show it, and then I think let's move on to the manual clustering thing, and then also I don't know, how can I stop share? Okay.
All right, anybody has questions for us so far? Something that you didn't understand or?
I have a question.
Mm-hmm.
How do you know that, I guess it relies on the assumption that the neuron is going to behave the same, like, at all times? Like, how do you distinguish between two different, like a neuron that's just firing differently versus two different neurons?
Right, so that's I would say like that's one of the main caveats of electrophysiology and spike sorting, because that's definitely like neurons change their waveforms because of burst firing or other I mean any other thing that can happen in the intrinsic mechanisms like of the cell, right? So it's hard to separate. We try the best, but it's definitely like a problem. You may get like so here, like it's complicated to call it cell. We usually call it clusters, and usually a cluster can be two cells. It's hard to distinguish. I'll show some. We are going to show some problems and some ways to go around it. But yeah, that's definitely a problem. Any other question?
What do you use for spike sorting? What's the name of your software?
We use Kilosort for the automatic,
Okay.
and then for manual we use a software, a GUI that is called Phi, and I'm going to show the Phi for you guys very soon.
Why are you not using the same one?
So Kilosort is for automatic. So it's the automatic
it's
part.
just that, okay.
Yeah, it does the steps that Soyoun showed. It filters and then like remove the spikes and then makes the spatial comparison, temporal comparison, and then we need to go and do a manual curating, I would say.
Okay, thank you.
Okay. Should we make a quick break? I guess this class today is going to go short, but if you guys want to make a break now. Yeah, okay. So we do,
Okay.
How long do you guys usually
coffee.
do?
We do like
5
minutes.
Okay.
Yeah.
So back in 11:14. Yeah. Is people back yet? I think I'll wait a little. 2 minutes more. Okay, so going back to the spike sorting, we wanted to do this second part to be very hands-on, but it's kind of complicated because the software we use right now, it's dependent on your computing having the right graphic cards. It doesn't matter which graphic cards or if it's a very good one. Apparently, like I'm having troubles to run on my laptop even, and it has like a good graphic card, and I'm going to show from my computer in lab. So I'll be sharing my screen from the team viewer, and I hope you guys can see. Let me see, I'm gonna share now. Can you guys see my screen, everyone?
I can't see it.
You can't?
Black.
I can see it.
Oh, now we see
Now
it.
it works now.
Yeah.
I can see it.
Okay, so I think I'll have to go slow because the team viewer is already slow for me. So I guess here in the Zoom is going to be even slower. Okay, so the data I'm going through, so I'm going to show this spike sorting, and the software is like a very recent, like they developed last year. The script, it's called Kilosort 2. It has some features different from Kilosort 1, but the principles are the same as Soyoun was explaining, and even if you don't, the goal here is for you to learn like how to differentiate like a cell from something that looks like a neuron from something that is not a neuron, and the main caveats also of this technique, not only the advantages. So maybe later in your research career, you may do spike sorting with other softwares, and I want you to keep like in mind what are the points of how do you differentiate cells. So here, what I'm showing is a recording from both striatum and hippocampus that we did in our lab. So in purple is striatum, and in blue is the hippocampus. We didn't have many cells, so I'm going to give a zoom here. Let me see how I'm going to start this. So we didn't have many cells on striatum, so I'm going to remove this from visualization because it will get better for everyone to see. Also, this data was collected by Adam that is here in class, I think, still. Yeah. So what we have here, we recorded using a silicon probe, as Soyoun was showing. The silicon probe is that design that has like looks like a fork and has the channels on the fork, right? So each of these groups, I'm going to put them in different colors now. Wait a minute.
Sorry, Ellie.
Yeah.
The striatum spikes or waves, those you designated them as striatum. Like, how did you do that? I kind of missed that.
Oh, sorry. So yeah. So we have here we have two sets of channels. One set we aim for striatum, the other one we aim for hippocampus.
Okay.
so the cells that I get on striatum, so the cells I get in the set of channels, I know that should be coming from striatum. So that's how I can differentiate them. So I'm just putting different colors here. So you can see each different color is a different shank. So you see that they behave kind of similar, but they behave more similar among them because they are in different parts of the brain, right? Oh, this is very bright. In a minute. Okay, so we have this. So we have 5 shanks. They are very close to the cell bodies. So what we see here, so we have the raw data. And stop any moment to ask questions, okay? Feel free, this is supposed to be more more hands-on, so I hope you participate a lot. So here, I'm going to give a zoom in one of the shanks. And you can see that there's like these high oscillations, but I don't know if you guys can see, I don't know if the quality is good for you as it is for me, but you can see these spikes here. You see this fast deflections that happen in the signal. Maybe these are too small. This one here down. Do you guys see this? This is definitely a neuron that is happening. Can you guys see this?
Yes, absolutely.
Yeah, so like this is a cell. So you see like there is this high deflection that happens like in some of the channels because it's not supposed to happen in all the channels. If it happens all the channels, then there's more here. Then it's likely noise. So you see there's some spikes and then they're going to happen. So our goal is to detect those, right? So for this, we follow those steps. We remove the slow oscillations, and then you're going to have just the high frequency. A spike happens, you guys may know, like in what, 2 milliseconds. So it's super fast. So when you filter, you remove this slow variation, but you get the fast spike, right? Actually, I saw something here and I want to show you. So you see these high events here. This is like very common in hippocampus. So it looks like it's noise, but it's a massive firing of cells. And so we call them, those are the, I don't know if you guys ever heard about them, but they are the ripples. And you can see the spiking happening on top of them. Here, can you guys see? I'll even remove the white shank.
Eddie, can you like move your mouse a little slower? There's a delay between your speech and your cursor, your mouse.
Okay. Okay, thank you.
And if you move it too fast, like it's too confusing. At least for me, and maybe my connection is too weak, but.
No, but good to know, definitely happens that. So I gave zoom now. Can you guys see this? Yeah? Yeah. Okay, so here we have like this very high oscillation. I'm going to remove the amplitude so it looks better now. So can you guys see okay now? Does it look better? So you see like these deflections that happen here are gonna show in the top channels first. So these deflections are high here and low in the other channels, right? And you see that the firing happened inside these events of like very high frequency. And you see how the amplitude of these events here, these are like slow oscillations. Their amplitude is very high, right? So what happens is because a lot of cells are firing together. So one of the problems that happens here is that many cells are going to fire at the same time. When your cells fire at the same time, you're going to have waveforms that look the same. Or even worse, what happens sometimes is that two cells that fire at the same time, they're going to sum their when you record, you're going to have a sum of the voltage. So what you see is actually like a third cluster. You see what I'm saying? That's one of the problems that Michelle was, one of the question from Michelle is that sometimes you can't distinguish them. Like most of the times you get very well, but most of the times you can't.
Could you
Sorry,
give an example? Sorry, could you give an example of one summing? I don't know if that's really hard to find or.
I could. It's hard to find, but I can. Let me see if I find here. I got some slides with the examples. Okay, I can try to draw actually. Would that work?
Yeah, that's fine.
Yes.
Okay. All right. So
just trying to visualize what it would actually look like.
Right. So what do you have? So normally you have like a cell, let's say you have a blue cell that is going to look like this, right? And then what happens, you have another cell, cell B, and the cell looks like, I don't know, this. They look very similar, but one has a higher amplitude, right? And you can usually separate them because they don't happen at the same time. If you have a stimulus and these two cells are recruited and you have just one channel there, imagine you have just one channel there now. I'm gonna drop my hand. So imagine like you have a tiny electrode and you have the cell here. Let's imagine a pyramidal cell. So imagine you have two neurons and they emit like a, so this is the red, no, sorry, this is the red. And this is the blue. So it happens like this one is farther away, so the amplitude is lower, it's lower, but they don't fire at the same time, so you can distinguish the waveforms. Then imagine that like they have a common input coming from another cell and make them fire together. So what happens now, like now these two cells, these two action potentials are going to sum in your recording. So you're going to have, maybe it's like, you're going to find something like this. Like even bigger because you just sum the two of them. And then you find this like cell that is super big and fire every, I don't know, very low firing rate, which means that it may be just two cells that sum together. This is a hypothetical scenario. Like sometimes what you find really is like, you find a cell that it does like something. I'm gonna draw another color here. You may find a cell that it fires, you may find a cell that is like two peaks. That happens a lot, especially if you go to record in a brain region that has like a higher firing rate. The cells that happen so close together that you get actually this, you don't get a nice separation. Is that okay? Make sense? Okay. I'll stop sharing my screen. Okay, so we run kilosort on this. Kilosort is going to take advantage of the spatial components, right? So we know that like the cells in the recorders on the same shank are supposed to have like action potentials that spikes on different channels. So it takes advantage on that. And what we get for your cluster, you go to the manual curating of spikes. So this is the main software. This is the file, and I just load the data here, and I'm going to explain some of the, so there's a lot of information here in this window, and I'm going to try to show the main important ones, okay? So first here we have the list of clusters. So these are all the clusters that the automatic, so again, we have automatic spike sorting and the manual curating. So now we're manual curating. Before I did the automatic clustering, which did all the process that Soyoun explained. And now it's showing to me the potential cells or potential clusters that may be cells, right? So we have a list here of clusters, all the clusters that came out. So here I'm going to click, show the first one. On top.
Hey, Ellie?
Yep.
Sorry. So what happened between kilosort and this? Did you bandpass filter that data as well? And then
Yes.
cluster it? Okay.
Yeah, so I basically, So the kilosort is going to filter the data, is going to extract the spikes, exactly like Soyoun showed, and then it's going to, it does a little it's a little different, but it also like relies on feature extraction. So the features of the waveform. So how the waveforms look different, right? To separating them in clusters.
Do they use PCA, or how did they do that?
They use one that is template matching. So yeah, that's a little bit different. So so template matching is just like, it's exactly the name. So you create these templates and you see like how do they match to them to like make this spike sorting. So you have both the spatial templates, which will be like, look here on where's my pointer. So this will be like a spatial template, and then you have the temporal template also. So this is good when you have, so this method of template matching, it works very well if you have high density of channels around the cell. Okay, imagine like we have a lot of, I'm gonna find another example here. So imagine you have like many channels, and then you can do like a very good template matching because it basically looks like a figure, right? So I'm gonna show here one example. I'm gonna share another screen with you. Here. So imagine like we had all these channels, like 200 channels around one cell, and the cell fires. This is what you're going to see. Like the channels closer to the cell is going to be higher, and then like reduce. And then imagine like we have this cell close here, right? Can you see my pointer or no?
Yeah.
So you have the cell around here. So imagine we have a cell now that is on the top left, then it's going to look like this. So you can separate the cells because they have a spatial distribution different, right?
Mm-hmm.
But imagine like now you put just one electrode or only 4, so it's not so good to separate. So it's not recommended to use template match in that case.
Okay. Gotcha. Thanks.
Yeah.
Sorry, I have a question.
Yeah.
Please correct me if I am wrong. You mean that the software distinguish different clusters according to the distance of each neurons from the channel, yes? For example, if a neuron is close to a channel, we can have the more actually amplitude, yes? And for the far neuron, we don't have. Yes?
Yes.
So the question is, what happens if the farthest neuron fire more power than the closest neuron? How distinguish between, I mean, the power can, I mean, you know, when farthest neuron fire more powerful than the closest neuron, maybe the software finds that this is the nearest, I mean, neuron. I don't know.
Right. No, that's a good example, and it can happen. It can happen. That's definitely a problem, and to overcome that, some people use also the temporal loadings, right? So a neuron that is far away, its action potential is going to arrive in different times in different channels, right? Even though it's firing stronger, the speed of the electrical field is not different. It can't change. So what happens is like, I'm going to give a zoom here on the waveform view. So I'm going to increase this window. Very slow. It's not increasing. Wait a minute. Okay, there you go. So you see that like the peak in each cell, in each channel is going to be different. So you can, that's the temporal template matching. So then like you can solve that too. Then you can know if it's the other cell because the temporal profile on the channels are different. Does that make sense?
Yes.
Okay.
Yes, thanks.
All right. Okay, so here we have all the clusters that came out. This is the parallelogram view. So this is it's basically a histogram of the time shift spikes. Okay.
I don't think the.
Oh,
I
I see. I'm sharing the wrong screen, right? Okay, there you go. Yeah, I was showing this example, sorry. Can you guys see the waveform view? Yeah, okay. So here's the parallelogram. Can you guys still see my pointer? Okay. So that's the parallelogram. It's basically a histogram of the spike time. So you get this spike time and you shift in time, right? So you shift like 1 millisecond, 2 milliseconds, 3 milliseconds, and you build a histogram of that. So that's basically what it is. If you guys remember from, I think everyone took molecular cellular neuroscience, right? Like, you know that the neuron can't fire because of the voltage sodium, voltage dependent sodium channels, right? They can fire in a specific time window. So you have to look to that and see here if the neuron is not violating that. So if the cluster you got is not violating that limit, which is like the 1 millisecond, right? Or 1 millisecond is it turns out to be the extreme limit, but usually a cell has like 2 milliseconds of 2 to 5, depends on the cell, right? We have the parallelogram view here. So again, this is time here and the counts of spikes. We have the waveform view where you can see the channels from the shank. So when I click to select the cluster, it gives me this channel. So I'm going to show the raw data. So this is the raw data, how it looks like. So you have the channels with the spikes, right? So this is one shank, has like 12 channels. This is one important because once you look here, you want to see if the activity that was detected in this cluster looks like a cell. So what are the examples? Usually you see this like fast peak happening. I'm gonna give it a zoom. So you see this fast peak and then the, what do you call this low wave happening after. There's different cells, they have not only negative, you can have cells that have like positive spikes. And also what you want to see here, when you have many channels, it's that the channels look different. Because if they look the same, it's probably like noise. Another way to spot noise if you don't have these channels is to look if the negative and positive activity are the same magnitude. So if you see a fast spike, I'm gonna draw this. So so if you see an activity so the cell usually is going to look like something like this, or like a positive spike like this, or even like just negative fast. But if you get something that is like, sorry, I'm gonna draw again. So if you get something that has the same magnitude of positive and negative, it's probably an artifact. So that's another way to spot. You can have so like you may, let me share the other screen. So you may be here and you find you find only 2 channels that have like activity. So it looks like maybe a neuron, but you see the activity is equally positive and negative. It's probably an artifact that those 2 channels are getting. Okay. The other important part of spike sorting, I always look at the features. So like I have the features here, which are like the PCs, like the principal components, right? So for each shank, for each channel, we get like 3 principal components. So you guys can see the one I'm moving right now. So these are the principal components. So I'm going to explain this. The first row is the first principal component against other components and against time. So the first row and the first column is the first principal component and this component variation in time. So you see that sometimes it goes up, goes down, goes up, goes down. And the second column is the first component against the second, the first against the first, I think, the first against the second, first against the third. And then you have all the combinations possible. So the goal here is to see the cluster, and if you see anything, for example, if you see that your cluster has a lot of noise, then you may clean it. So for example, we can try. You see here that my waveforms are not that great. That's because we're recording the animal freely moving, so you can get like spikes when the animal it's crunching food or scratching his ear or something. So we can try to clean. So the way you clean, you just come here and you try to cut this cloud of points. So the blue is the cloud of points we are seeing. The white one is just like the background noise they put here for you to have a reference. So what you can do here is try to cut this cluster and see if you can find another cell. So usually like you select here and then you cut, and it's going to show you 2 different clusters that you did now because you cut in half, but then you see that so I'm gonna look here, we're going to go over this in more detail, but you see that they have like very close, they are the same cell because they have the same amplitude and the same autocorrelogram, right? So it's not a good idea to split the cell, right? They're just showing how to cut. So you may try to clean, to cut like the borders like this, and you clean the cluster. So usually when you're doing spike sort, like don't waste much time cleaning a cluster, trying to make like a very clean cell, and unless you have like a very good reason for it. Most of the time during spike sorting, you are not getting all the spikes of a cell anyway. I've seen some studies, at least I saw this poster in SFN like some years ago, that you have, they did just juxtacellular recordings. So they have like a pipette very close to the cell, and this pipette was glued to a silicon probe, so they could record and do spike sorting at the same time that they do the juxtacellular. And they basically find that the best case scenario, you get like 90% of the cells, but most cases you get like 70 or 60% of the activity of that cell. So it's not a perfect technique. It has its flaws. So what my message is that sometimes it's not worth to waste so much time trying to clean a cluster. Just like get what you got and do your best, but it's just better to, I don't know, do more experiments, for example. So okay, so this cluster based on doesn't seem like I can separate in any further clusters. Looks like a cell here, and the correlogram looks good. So I'm going to assign a label that is a good cell. Okay, so it's going to turn. You're going to see here in the cluster view is going to turn green. Now we have a good cell, and now what you do, and what's usually like very time consuming, is go through all these clusters and say which ones are good and which ones are bad.
So okay, look how this cluster made. What's the difference between one and the other?
Say again, which clusters?
Those clusters, like, so there's different type of clusters you are basically going through to find which one is the most
right
faithful to the situation, right?
Yeah.
What's the difference between one and another? And
well, the difference.
Did you write the code that would create those clusters, or did the software do that for you?
No, the software does that for you based on the which channels they like, based on the channel activity you get, and based on the time activity, right?
So what are the different types of clusters then?
So like this is where the first cluster I show you, right? So here I'm gonna go to my stream, right? And you can see that, like, you see these points in blue here on the right of my screen. I don't know if it's my right or your left, because I know that Zoom mirror the camera, I think. So you see here probe view. Yeah, so you see the like this cluster, for example, it's on this shank. So we have here the shank that I put on the hippocampus, right? So like this points here, this column is like one shank, this other column is another shank, and the blue one is where this cluster that I'm showing you, it is. So like you see that it's happening in this shank, and when you look at the channels, you see that the cell has this spatial pattern, right? So now I'm going to go to another cluster, and what you see is that this cell has a different parallelogram view. It has a different, I'm gonna put the average. It has a different spatial here, and it participates, and it's actually like in another shank. So it's extremely right. So that's how you separate them. The automatic cluster does that for you. And then now we just curate to see if they're different, if they're potential cell activity or not. Does that answer your question?
No, but my question was not phrased correctly. It's in your feature view window.
Oh.
What's the difference between one and the other, and how do you select like one and the other type of cluster separation that we can see only in the feature view? And how do you select one that is correct? Because that's the type of difficulty that I have when I spike sort. So
okay,
touch up on that a little bit.
Okay, so okay, I see your question. And how do you do your spike sorting? I use.
So I have one. You see what I'm talking about when I say your feature view window?
Yes, I know here, right? Because sometimes you have like different clusters here, right?
Yes. So how are these designed, and how do you what are criteria that make you think that you got a good one for doing your separation?
So usually when I see like a cloud like this, just a single cloud, then I believe like it's probably like just one cell. Like there's no clear separation here. Like let me show you like here.
Between blue and gray, right?
Exactly, because the gray is just the background. So like the blue is the cluster that is being shown, and the gray is the background activity of those channels. So it does the feature extraction, like the PCA in all the channels, and then it show you like the gray is all the whole thing, and the blue is your cluster that you're looking right now. So you see, like it's hard to see a separation, right?
Yeah.
Here, but I know what you're saying. Like sometimes you have like 3 clusters, and then like it's sometimes you have like a banana shaped something, and then like you don't know if you cut or not, right? That's a trick thing. Hopefully we are going to go through one of those, but I can show you one example too. I have a slide with an example, and I can, let me see. Okay, I found one slide here.
So we're still on a kilo sort.
Okay, what about now?
Cool.
Nice. So yeah, this is the thing you're saying, right? Sometimes you look in your feature extraction. Here you see like 2 clouds, and they look very separable, right?
Yeah.
Is that what you're talking about?
Yes.
Yeah. So then like this is a nice example, because you can separate clearly. Like you can just put a balloon around here and like cut this cluster and separate them. But sometimes it's complicated.
This is when you're lucky, but
yeah, exactly.
This rarely happens to me. So what do you do when it doesn't happen like that?
When it doesn't happen like that, and like when it's very close, and you're like, sometimes it may be the same cell that has different features. So like if a cell is bursting, if a cell bursts, the next spikes on the sequence of burst. So let's say like you have 10 spikes in, I don't know, 10 milliseconds. The last spikes are going to be smaller, because like the cell use a bunch of sodium channels already. So like you can open all of them. So everything is going to change. So sometimes what you have, instead of a nice cloud of points, you have like a banana shape or something happening like that. It may be the case of your cells, and then you can assign safely and say like, okay, this is a cluster, probably a cell. if you don't think that's what's happening based on the looking at the waveforms, for example, then what I do is to assume is a multi-unit activity. So like that cluster got more than one cell. So that seems like not good, right? Like, oh, spike sorting I'm doing because I want to have different cells in different clusters. But for some analysis, it's okay to use multi-unit activity. So if you're trying to predict the animal position, if you're trying to predict like if the animal is scratching his left or right here, like the multi-unit activity may add information. So in this case, like you can use those cells too. Let me see, let me get a good example of bursting. Soyoun was showing, just a minute. Sorry, I need to find it. Here. Yeah, so like this is an example of bursting. We may find during your hands-on spike sorting, but I'm going to show here first so you have an idea. So sometimes what you get is like this cell, this blue cell that fires like, that has a big spike, and the red cell has like a smaller waveform, right? And when you to compare them, what you see in the feature view over time, you see this pattern that like you have the blue cell constant, and you have like this red cells happening sometimes like in very quickly. So then what happens, that's probably like as it's either a cell that is like it was splitted, probably like two cells that were wrongly splitted, or it may be the burst that is happening. So I'm gonna show the next slide. So you may have, this is the burst. So like you have the first spike here is the biggest one. And then when you have the subsequent bursts, you see that the spike changes a lot. So you may get a cluster with the last spike, which is a 6, is totally different from the first one, right? So how can you solve that? It's complicated, yeah. So what happens sometimes is like you can do this plot, you compare the two cells and you see this, maybe the burst. And the main thing to know if it's a burst is to get the parallelogram views. So I'm going to give a big zoom here. Oof. So, oh, sorry, stop sharing. Are you guys asking questions in the chat too? Or, okay. Oh yeah, great. Claire shared a link. So yeah, so one of the ideas is to you put the parallelogram. So you have the parallelograms of the blue cell, which was the first spike, and the red cell, which was the subsequent spikes. And you see the parallelograms are very different. But when you compare both, you have this cross parallelogram as we speak. It's like you get the spike of the cell A and the cell B, and you do like a cross histogram of those spikes. So what you see here, for example, in this upper right plot, is that the red cell usually fires after the blue cell. So if it was on this side, it would fire before, right? But it fires after. So this is exactly the opposite of what we see here, right? So here is showing that like first for every spike you see in the red cell, usually you have a spike from the blue cell before. Does that make sense? So when you see this inequality in the clusters, usually is a burst. It's like you see that the second red cluster happens after the blue most of the time. It never happens before. Never happens is too strong of a word. It's like it's unlikely to happen before. So usually this means that the cell is it's a burst. Does that make sense? People have questions about this? No?
Could you please repeat it again? Sorry.
Say again?
Yes.
Oh, about the parallelograms or?
No, about why, for example, sorry, I missed it, that which of these shapes is representative of the bursting?
Oh, okay. So the red one is supposed to be the, it's likely to, it's our candidate bursting, right? So when the cell fires the first time, the spike is very big and like very nice waveform. But what happens when you have a burst is like the cell can't open the same amount of voltage gated sodium channels, so the waveforms change. And the whole point of the spike sorting is to separate cells based on their waveforms, right? So when this bursts happen, you change, the cells get in different clusters. So one way to identify is to either look at the feature to see like how the features are, you see here, kind of correlated because they happen very close. I can show you an example of when the features doesn't look like they are related. You see, like this looks like a continuum, right? Like this is in time. So the x-axis is in time. And another clue that it's probably bursting is to look at the cross-correlogram between these clusters. So the cross-correlogram is the same as the autocorrelogram. So you count the amount of spikes as you shift in time. But now you're counting the spikes from A as you shift in time B. So what you see is that usually the cells in B fire after the cells in A, and other way around. Okay.
Thank you.
I have a question about, I might have missed this, but what is the time scale for the lag you're looking at?
For the plot here of the features?
Yeah, for the correlograms, just like
Oh,
how
sorry.
big of a time lag would it be before you're just considering these two signals like don't have anything to do with each other?
Right. So this window by default, you can change that on the software usually, but by default the correlogram is like it shows 50 milliseconds.
Okay.
Let me check here. Is in the correlogram view. Let me see. Set window. Yeah, so it's 50 milliseconds and each beam is 1 millisecond. So here the center is 0, so you have like 1, 2, 3 milliseconds you have the spike. So 3 milliseconds is very close. And by this by this pattern that you see that is not, it's not even, right? Like you have just on the right side, it's probably like bursting.
Okay.
Yeah, because it's the subsequent spike of that cell.
All right, thank you.
Yeah. Any more questions about the burst? I just wanted to go over one more example and then we can do some hands-on spike sorting. Okay, so let me see. We can share these slides with you. So one other problem that we may have, it's the drift. So imagine like, you remember how the silicon probes look? They look like a fork, right? But you know, the fork, it's let's say like it's rigid, and the brain is like, what? It's a jello, right? Like the brain is very almost liquid. So when you have like a fork inside a liquid, it's the same. So the idea of, imagine like you have a fork inside a jello and you're driving a car, right? What the fork is going to do is going to be moving around the jello, right? So that's basically what happened when you're recording with silicon probes in the brain. As the animal moves, if he moves too fast and hits the implant on the wall, or like the brain is going to move relative to the channels. So what happens is you have these drifts. So as I'm showing here, like this is a very high density channel, is the neuropixels. So you have like a records here, and each dot, so you have 1 hour recording and each dot is a point, is a spike, right? So you have here this very high density spiking cells here in black. And what you see is like when the animal, probably when the animal moves, or like the electrode move a little bit, you have this like shifts up or shifts down. So this is another problem, like one spike that is in a set of channels may change to another set of channels. And then you lose, and you're going to have like 2 clusters that is the same exactly cell, right? That's a problem, like when you're doing behavior, you may have cells that are firing to the half of the behavior, and then you can't use for the whole task because, I don't know. So like this is a problem, like people are kind of able now to fix this problem with the drift correction, which is a feature from the KiloSort 2, right? So what they do is basically get this, they get all this, like they get a window of time, let's say 10 minutes, and they get like how the spikes are distributed. This is like a very simplistic view, right? I'm just explaining to you. So if you have more questions, we can talk another time, or I can, I mean, you guys can also look at the links that Claire sent. Is very useful to understand how KiloSort works. So when they get these 10 minutes windows and they calculate the similarity between them, so then like they can adjust this drift, right? So what they do when they correct the drift is that you have like a nice smooth and straight, or let's say recording, and then you can track the same cell over time, right? So we're going to see some examples of that on my recording as well that I was showing before. So here, for example, which screen I'm sharing? Okay, TV. So for example, this cell, you guys see the over time it fluctuates. All right, so like in other spike sort algorithms, maybe this will be like separating different clusters, right? Different cells. Okay, so I guess that's most of it. So we can make some, you guys have any questions before we go to look at more clusters? No? Okay, so yeah, one idea of like spike sorting is just, you just have to do a bunch. There's not much secret of like how manually curate. Like if it doesn't look like a cell, it's probably not a cell. And if you have like mixed feelings about a cluster that like, oh, I'm not sure, then it's better to like just not use it or assign a multi-unit label, right? Okay, so here I'll do, let's say 3 more clusters, and then we start making a voting. How do you guys like them? We can, I see that here in Zoom, if you go to participants, you guys can select yes or no. So we can make like a voting. All right. So I'm just gonna go for some clusters here. So this cell, this cluster here, for the first side looks like a cell. Doesn't seem like I can separate on the features. The waveforms are not that big. It's not that great. And I can look at the average, and you see that there's kind of like 2 peaks. There's probably like 2 cells that fire very close together. So you have this cluster, right? So what do we do with this? I would set up as a multi-unit because of these 2 spikes. Even though it's a very good, it has a very good parallelogram. So like the more you see like this clean here in the middle, it means that it's not violating the refractory time of the cell. The better, like the more clean the cluster it is, I would say. Okay. So this I'm gonna put like assign a label of multi-unit. So it goes automatically to the next cell. Now the next cluster, sorry. So this cluster, first you look at parallelogram, you see that there's a lot of activity. So these bars, they are a little bit offset, but this bar here, can you see the lines? It's very thin. These lines are to tell you like what's the refractory time. So I think this is 2 milliseconds. I don't remember, let me see. Refractory period here. So yeah, it's 2 milliseconds. So you can change that line. So I can put like 5 milliseconds, and you see that the line increases. I'll keep in 2 milliseconds because I like that one. Okay, here now we see the spike looks like a noise. The waveform looks very bad. I'm going to look on the average, and you see that the waveform looks the same in all channels, and it's violating a lot the refractory period. So I just assign as a noise. This other cell also violating a lot, this other cluster violating a lot the refractory period. So you see this very high here. And the waveforms also look all the same through the channels, probably noise, can't be a cell, okay? This is another one, is another noise, another cluster that is a noise, high refractory period, and all the activity in all the channels look the same. This one looks like a neuron. So you see that the autocorrelogram looks very nice, and it has some, the activity is different between channels. If I remove the average here, you can see that it goes changing. And it doesn't seem like it can separate, so it's a good cell. I'm going to assign the label good. Okay. Let's do some. We can make like 10 or 12 clusters. Okay, so everyone can access the, if you're going in participants, you can vote yes or no, and I can see here when you guys vote, all right? So, I mean, that's the best hands-on I can get for you guys. So what do you guys think about this cell? Is it a cluster, or is it a spike or not? Yep, very good. Majority vote yes. Yeah, it is very likely a single cell. One important feature I want to show to you guys here too is, like you see here in the feature view, you have this changing feature, probably like because of the drifting that happens during the recording. And you see that also the density gets slower, the density of points get lower through the end of the recording. It's probably the cell stop firing, or like the cell is dying, or it may be you're just losing the cell by the end of the recording or something like that. But yeah, it's a good cell. So next one, what do you guys think about this one? Can vote again. All right. Yeah, so this one, it may be tricky, but it is definitely has like components of a neuron here. It definitely has noise. So you can see here the middle, it's violating the refractory period, but is still a cell. So I know that in the spikes here, they look similar, but they are changing over channels. But yeah, look, it looks terrible. It definitely would need some cleaning. I wouldn't put as the best cell I have in this recording, right? But you can see here the features. The spike happening. There's a lot of, like, it would need like a good clearing of these spikes, of this noise around. Yeah. Well, so yeah, this autocorrelogram that looks like it has a burst here and decays, and we know that we are in hippocampus. Is probably a pyramidal cell. In hippocampus, they are very common to find like this kind of autocorrelograms. So different brain areas may have different correlograms. So it's hard to find like a cell like this in striatum or like in other cortices. So it's a good cell. Okay, let me go back here. So what do you think about this one? It is definitely a cell too. It has like noise, but you can definitely assign as a cell. The waveform features, it's more or less all over the channels, but they are different. They are not the same. So it's definitely a good cell. What about this one? Right, this is a good cell too. What about this second one? Yep, that's a good cell. What about this? This is a new cluster. You guys haven't seen. I think I didn't show before. What do you guys think? Okay, mostly green. All right, I see some reds too. Kind of mixed feelings from the class. It's mostly a cell because the parallelogram looks very good in the mean that like it doesn't have spikes, and it's like you have this smooth transition, which is probably like a biological effect. It's a biological process. It's not like some noise, right? It's if you guys have questions about clusters, you can like just ask me. Like, oh, I thought this wasn't a cluster because of this and that, and then we can talk about it. It has only one channel, but this can really happen. Like you have this positive waveform that is happening in like one single channel, just like maybe that channel is better oriented with the cell or something like that. And the spikes are not great. You can see that like some part of the recording we missed the spikes, but still it would save as a cell cluster. Okay. This is a tricky one, but yeah, I would assign as a cell. What about this? Okay. Yep, very good. It's not a cell. You can see like they're pretty much the same all over the channels, and it doesn't respect very well the refractory period, so we assign as a noise. So yeah, you guys got a sense on how this work. I can show you like many other examples. Oh, this one is a good one. So I see you guys are very well trained now. This is definitely a cell. The problem here is that we have this activity you see here in this is one of the problems that like Noeli was talking about. So we have here the activity in one period of time. You have this like weird activity, and the cluster doesn't look like very good to separate. So what do we make of this? At this point, like it's important so you know what happens during your experiment. So I know that like this animal was recording for sleep for many hours, and then we did the task, and then we did more sleep recording. So this probably happened during the task, right? So what happened during the task, like the animal is moving around, may hit the head, change a little bit the electrode, and you may have like one cluster going to the other. And like this looks exactly like that. I don't know if you can see with the good resolution as I can see here, but this cluster looks like it was a cluster that was like going up here, or maybe was even high here and just drop, and like the waveforms look very similar to the cluster that is actually the cell that we're looking, right? So what we can do, I don't know, like at first I would say like it's a multi-unit. So you can try to cut the cell and see what you get. So there you go. So now you see like a separation. When we look at the parallelogram view, we see that the one spike the one cluster against the other, you see there's it looks like one is like the burst of the other, like the subsequent spike, because it has this peak here. It's not like the example I showed you before, but it seems like, so it doesn't seem like the cut I made on the cluster was very good. So this is one of the problems like Noeli was pointing out, like sometimes you get this kind of stuff and you don't know where to cut. So what you do? I would say keep trying to cut until you get like different cells, but don't spend much time on one cluster. Really, don't spend like just assign as a multi-unit and that's it. Like this is not the autocorrelogram of cluster A and B looks very different, looks like a different cell, but you see that they have cross-contamination because it seems like the cluster in B has some components from the A, like subsequent spike. It's complicated to cut here. We have this one. If I try to cut during the temporal components, still the same. The waveforms look very similar. It's just like a hard cluster to tell what's going on. So I would assign like multi-unit. Okay, so noise. So what I do usually, I go, this is another one, but that's one is, it's kind of clear where to cut. If this is not, so what you're seeing here, if this is not drift of the electrodes, this is probably choose different cells. You can see like a clear, this one is a good, like you can see a clear cut. Oh, so look at them. So it turns out, this is the example I showed you before, right? You guys agree? This is the burst. So I see the cluster A was it, and the cluster B has a very nice close autocorrelogram. And then when I do the cross correlogram between these clusters, you see that cluster B happen most of the times after cluster A. And these two clouds I got on the feature view is just because the second spike has a very different waveform. Has a smaller, or so you can see here when we compare. Can you see? The red one is smaller than the blue one. It's because of the burst. Probably they are the same because they have the exact same spatial distribution. Okay, so we don't cut. I cut the cells. So why we had two clouds? So we don't cut those cells and we assign a good. So like that's a good cluster, probably a single cell. All right, so what I do usually when I'm spec sorting, I go over all the clusters doing that. It may take you like, in the beginning, it take you like hours. It may take you the whole afternoon to do. But as you train like to get fast on this. And like, you look at the cluster and like, oh, this is good, this is bad. This may be weird. I'm going to try to cut here and there. So you just assign like, oh, this is good, this is good. And you go like very fast, like, oh, this looks good, this looks good. And you go cutting, you go very fast. Like, don't worry in the beginning. So I do this first pass, then I look like, oh, it looks very good. What does it look like? Noise, so like this noise. And I go very fast. And then later on, I select only the ones that are good and then I spend time on them. So you don't spend time on noise, okay? Once you see like it's probably noise, just throw away. So after I go through all the clusters, I go back here and then I go like cutting. I see if I can merge clusters. So for example, I'm going to see if this cluster and this second cluster, if they are related. So this cross correlogram has nothing to do with each other. You see there's like kind of a straight line. So I don't merge them. This is another step, like after you assign all of them to see which one is good or not, you have to merge cells, because what happens during spike sorts that you may over split clusters. And that's better than like over merging, because it's harder, as I showed you, it's harder to separate one solid cloud than merging them, okay? Like look at the cells, if I merge them, they are going to look exactly like one single cloud. And then like once I merge them, I can't separate them easily, okay? Because they are going to exactly look the same. Like there's no easy line where I cut them. So I'm not going to merge. Then you go over the other cells and you see like which ones are related. So like, look at this cell. This is a good example. So this is a good example of merging, okay? Okay, so I have the cell. You look at this other clusters and you see that like their correlogram are similar and their cross correlogram are similar. So probably like these are the same cluster that were splitted, okay? As I told you, it's better to oversplit than overmerge. So you look at the spatial distribution in the channels and they look like the same, the both cells, right? And there's this template feature view that plot one against the other. You see like they have like a nice combination here. Let me show you one that is not. Okay, let me just finish this and I'll show you a cluster that is not good to merge. So you see here the in template view, they are like all intercorrelated here in the features. Here too in the cluster, they're like basically the same cluster. So like it's safe to merge these two cells, okay? What's not safe to merge? Okay, let me. Like this cell is not safe to merge. Their spatial distribution may be similar, but like the waveforms are very different. And here in template feature view, you see that they don't correlate very well. So like it's not as safe to merge these two clusters, right? I can show one more. Like all these clusters, they may be the same cell. Like this is another cluster, they have the same distribution, spatial distribution. Look here in the template view, they may look like one may be related to the other, but you see the cross correlogram doesn't indicate that. It's indicated like they are different cells. So the correlogram of each unit separate looks different and the cross correlogram looks like totally different. So you don't merge. So when do you merge? You merge when you get like stuff like this, okay? And then you can do merge. So like I'm going to plot a lot of clusters together. So like this cell was a cell that was like oversplitted because of parameters that you set on the script on the kilo sorter. All right. So these parameters are fine-tuned. So it doesn't come like straight good for you. So it depends on the how your channels are distributed, the distance between your channels, where are you recording. It depends on many stuff. So it's usually like fine-tuned for each lab, like or each project. Okay, so you have here, so you have all these clusters, like 100% sure they are all the same cell. Now, maybe if you merge them, you're going to increase the amount of spikes that you have violating the what's the name again? The refractory period. Okay, but I'm going to merge them and see what happens. Looks very good. It looks like the refractory period is still the same. And now we have like cluster with a lot of spikes. Okay, nothing weird happened to the waveforms. They still look the same. So it's a good merge. Now I'll try to merge something that is not related, okay? So like these two cells, I'm gonna get one, there's more. So like these two cells, I look at these two cells and I decide to merge them. Now look at the average spike, look at the spikes and average spikes, and look at the correlogram of A and B, and then when I merge, what happens? It becomes something else. You have like noise here. It may look like, okay, like still acceptable, the refractory period, but then the average waveform now looks funky. So you see there's like this, it's almost like an oscillation because they are like, it's not like a real like fast spike there was before. That's for users to have an idea of when to merge and not to merge. And like more dangerous, like when I merge these two, look at the feature view. You can't separate anymore. Like it doesn't matter how I cut. Okay, I'm gonna cut now. It doesn't matter how I cut now, it like looks the same. Like it's impossible to separate once you merge. So that's why it's better to oversplit than overmerge, okay? Because it's easier to merge the clusters. I guess that's pretty much it. I don't know, should I talk about something else, Soyoun? No. Yeah, I think that's pretty much it. I hope it's been useful. And like, there's no really like big secret in spike sorting. You just have to do it, and you'll get to know what's a cell, what's not, you know. So I hope this semi hands-on was useful for you guys.
Thank you.
All right.
Thank you.
Okay, I guess the class ends.
Mhm.
Stop.